So, talking about just one page on virus on COVID. Funny enough, there's been quite a few people in the last few weeks saying they know someone who's had a COVID infection recently. But looking at the figures of people who have been admitted to hospital, there was a peak in May, but it tailed off in June, and there really shouldn't be not very many people around now who get admitted to hospital. Of course, one of the features of this series of this pandemic is that the virus is gradually getting less and less virulent, so we're more and more, or less and less likely to end up in hospital. So the figures I'm seeing, which are hospital admissions, are staying very low, but that doesn't mean that there are people out there who don't end up in hospital, which is the vast majority of us, or having a little surge in the country that we're not tracing. The government is no longer really following this information very closely. We've got cases which are people who, if you go into a hospital, you're immediately tested. So most of those people would turn up in cases, but everybody else in the country, we're not going to know who's got COVID because we no longer collect that information. We're doing fewer and fewer tests on the people that we coming into hospital and so on. So the take-home message is it's pandemic has become very, very mild in terms of the chances of you ending up in hospital. And it's the same safety recommendations. I think you should all have been, if you were eligible, you should all have been vaccinated now in the spring schedule. I also know that, incidentally, that we've now had licensed a dual COVID and flu injection, which I will stick my neck out and predict that's what they'll be doling out in the autumn of this year, but I could be wrong. I might be a bit early on that, but there is now a fully operational dual vaccine going to be handed out. We didn't, in the hospitals, we didn't get vaccinated with the spring vaccine, so they've moved that away from a large population who work for the NHS as well. So they're obviously not at all worried about how severe this infection can be. So that's all good news. Just if you still feel nervous, just pay attention to protecting yourself in the normal way and get yourself vaccinated. So I spotted earlier this week a groundbreaking research paper, which harks back to talks I've done years ago, where we looked at things like the potential for lungs to repair themselves. And there's actually been a lot done on that before the pandemic. And I must revisit that area because I'll bet you can now rebuild lungs in a much more effective way than we could back then. And I think I probably last talked about it 4 or 5 years ago, I should think. But this paper kind of is along the same lines. But this is actually something which looks to be much closer than replacing whole sets of lungs to actually being of some practical use to patients. It's not there yet, but it's very interesting. So essentially, if we pose the question, why do some people get aspergillosis? And obviously, a lot of people don't. Most people don't. We can think of various reasons. For some people, there is the odd person who gets CPA after a massive environmental exposure. I'm thinking of people who've stored compost in a sealed plastic bag and then opened the bag the next year and been enveloped in a cloud of spores. Now, most of those people will just have an allergic reaction or no reaction, but there are a few cases that actually made the newspapers years ago where they got an invasive aspergillosis. That's one possibility, but for the most of us, we're not exposed to that level of exposure. We know for things like CPA and ABPA, if you've got asthma, you are at a higher risk. Also, TB, and those infection scars can act as a point of infection for Aspergillus, and you'll end up with an aspergilloma and CPA. Bronchiectasis is also a type of lung damage that a lot of it comes from things like smoking, but also other environmental exposures, and that can predispose you. About 15% people with bronchiectasis seem to get an aspergillosis of some sort. With TB, it's a little bit less sure. Most of the work on TB has actually been done in Africa, and they've found, I've seen figures ranging from sort of 5% of people with TB that's recurring and is not responding to treatment, can about 5 to up to 15% of those patients, depending on how you define them, end up with an aspergillosis as well, a CPA, which up until now was probably mostly untreated because there's not a lot of, or there didn't used to be a lot of antifungal drugs available in Africa. But now the awareness is there, and we've got several people who worked with us over the years who are now working over there, especially in Nigeria and Uganda, places like that. There's more chance that those people will get the correct treatment. And also, if you have maybe even just a slight vulnerability caused by your genetic makeup, there are things called primary immune disorders, which are almost always found in young children because these things are quite severe, mostly quite severe, and those children don't live more than a few years quite often, possibly not getting to their teenage years. But there are much milder versions, and you can even live till you're relatively elderly. And it can even, because it's so mild, it's not detected until you're well on in adult life sometimes. So there are different levels of it. And also much more subtle immune disorders that you're mostly able to fight off infection, but there might be some specific vulnerabilities around fungus. I'm thinking of things like where cells that line your airways stick close together. The fungus, no infection can get through, but fungus is able to, because it's got these strong threads, these tubes that it puts out, they can maybe get through the layers where most other infections can't. So if that's a little bit weaker and the cells aren't stuck together quite as tightly as they should be, it could be only a 10% reduction, and it's enough to sometimes let the fungal infection, the occasional fungal infection, get through. So these disorders can be quite subtle too. Most of you will know, or some of you will know, that we do a lot of our research work with Manchester University and the Manchester Fungal Infection Group, NFIG, as it is depicted there. And Norman, who's going to come and talk later, works in that group. Now we've got through the restrictions caused by the pandemic, then we're starting to work again more frequently with the university. But years ago, I think we're talking about 2010, 2012, so right at the start of NAC, we started doing a study, or we got funding for doing a study of gathering tissue samples from, I think it was 100 people with CPA and 100 people with ABPA and 100, I think we even did SAFs. And because of the advances in technology, we can now sequence that entire genome of those few hundred people very, very quickly. And nowadays we can do it for about $400 and do it in a couple of days, which is an amazing thing. I don't need to go into it just now. That's easy. Analyzing the data, not so easy. You've got 3 billion base pairs to compare, which takes a long time. But we are beginning to see the results of that. There were a few papers published a few years back, I think, on vulnerability found in ABK, but there's lots more to come. And Paul, who gave us our talk at World Aspergillosis Day, in February hinted at the end of his talk that they'd found something very important, but they couldn't talk about it too much just yet. And I'm sure we're getting closer to the point where they were able to tell us more about that. They've told me a bit, but I can't say anything else. So I'm just going by what Paul has already said. So that's really good. There are genetic differences. They do seem to make us people with CPA and ABPA slightly more vulnerable to these infections, and that might be one reason why you get an aspergillosis infection and other people don't. So it could be genetic mistakes, genetic mutations that we wouldn't normally notice, but you've just got that slight vulnerability and it's caused an aspergillosis. Yeah, save for that slide. So people who get chronic aspergillosis have some of the same mutations. They found, I think I remember the mutation they found for ABPA patients. They found about 15 to 20% of those patients had the same mutation, and that accounted for 20 or 30% of infections in some way. They did tell us it was due to the airway cells supporting more rapid growth of the fungus. I think it was something like that. That's been out there for 5 years now. So, and there are more. So there are mutations in common between people with CPA, and from what I understand, at the time the comment was that CPA and ABPA and SAFs are essentially variations on the same disease. So what that's hinting at is there are mutations in common between all three. So the information we get about one gene is widely applicable across all these chronic aspergillosis cases. How can we use this information? So if we were able to, because we've got this new DNA mutation information, we can use it in a number of ways. Clearly, if we found that these genes make certain people vulnerable, we could start to screen out individuals who we think might be vulnerable. So maybe individuals who've got asthma. We might screen out young children and realize that they're potentially vulnerable to aspergillosis, and then we can protect them from aspergillosis in some way, maybe more than one way, maybe even something like an aspergillosis vaccine, for example, which doesn't yet exist, but I know there's work going on to develop those. Because we've got that information, that tells us something about the particular proteins that are mutated and how they change when they've been mutated, we can probably develop new treatments, which are not antifungal drugs necessarily, but they would be drugs that would maybe correct the specific issues caused by the mutations once we know what they are. This is very like what has happened with treatment of cystic fibrosis patients with the new, I think it's a triple of treatment drugs which are really transforming the lives of people with CF. These drugs compensate extremely adequately for the problems caused by the mutations, and that has been developed because they know a lot about the CF mutation. They've effectively, you could almost call it a cure. It's not technically, it's not a cure, but there's a lot of people with CF who are living much better lives on Trifecta, I think it's called, which is quite expensive, but I think it's available in the UK since a year or two ago. And we could also, as a final option, and all my training is in genetics before I came here. So this is the one that really appeals to me. Back then, we looked at a whole range of people who are ill because of mutations. And it was a dream that we might be able to go right back to the source of the problem and repair those mutations in those people, thus giving them a normal life back. And that remains a possibility, and indeed is the subject of this recent paper. It seems impossible. What have I said? Yes. So the task is huge if we want to try and attempt to correct a single mutation or maybe a few mutations that we've detected by doing a genome sequence. It takes a couple of days. We can do that now. If we find a mutation, how on earth would we fix it? First of all, there's a huge amount of DNA to get through to find the one base pair mutation, or just a few base pair mutations. There are 25,000 genes in the human genome, which is big enough, but there are 6 billion base pairs in every one of our cells. That's an awful lot of base pairs to go through. It's literally looking for a needle, a genetic needle in a DNA haystack. Could that be possible? It would be amazing if that was possible. And of course, even if we could do that, we'd have to do it in billions of cells at the same time in order to restore function to an entire set of lungs, for example. So there are huge barriers to achieving this aim. Why have I put this picture up here? This is just an example. If you're familiar with DNA, it has this double helix structure. DNA is found in your nucleus, and this is what codes for all of your genes. And those are the things that build your bodies when you're developing in the womb. It's at its smallest, it's a tiny, tiny double helix, which is pictured in this cartoon. That double helix is packed into the nucleus of every one of your cells by winding round proteins called histones. Those proteins are wound together into these, what would you call them? Spirals. And those spirals are also wound. Excuse me. I think I may well have had COVID recently. So we've packed, it's just showing you the level of information in a single strand of DNA. It's absolutely massive. How could we possibly correct those problems, those mutations? Well, seems impossible, but actually, our nuclei, our DNA, has natural mechanisms to repair itself. And all we need to do is learn about those mechanisms, how to manipulate them and use them. How do we make those mechanisms make the right corrections how we want them to make? These things can do it. A process called DNA editing, and this has been known for a long time. But how we make it do what we want to do is a whole other issue. And there's something called stem cells. So all of our cells originate from a stem cell. So when we were tiny, there were probably a few dozen original stem cells, and all of our cells during our development came from those cells. Those cells don't go away. See if I've got another slide. Yes. So those cells don't go away. So if this is our lungs. Our lungs are made up of multiple different types of cells. You've got your airway cells, you've got your parenchyma cells, you've got your alveolar cells, and so on. And they're listed down here, large airways. Ciliated cells line large airways. Basal cells line the bottom of these airways. They're sort of outer surface. Secretory cells are the things that make all that troublesome mucus. And lots of different types of cells. Some of these cells are stem cells, so basal cells and secretory cells. And a stem cell is capable of making all these other cells. There will be stem cells throughout your lungs that's capable of restoring every single cell in your lungs. And that's what they do. They sit there, and every cell in your lung is replaced roughly every 2 to 3 weeks. So even though we think our cells are, mine are 60 years old, they're not, because those cells are replaced every 2 or 3 weeks. Some cell types are replaced more quickly than others. Cell types that are lining airways, for instance, are exposed to the environment, so they're exposed to a lot of damage, so they are replaced more rapidly than other cell types that aren't exposed to the environment so much. So it all kind of makes sense, even right down to the little air sacs that make up the majority of the cilia of our lungs. So we can repair DNA, and using stem cells, we can replace every single cell in our body once we've worked out how to manipulate stem cells for our purposes. Okay, now we've talked about stem cells before, and in fact, I remember it being an example of how science can go badly wrong at one time. Oh, we're not going to talk about stem cells yet. We're going to talk about DNA repair. Okay, so how do we make gene editing work for us? Well, the natural system of DNA repair and editing that's present in all of our cells normally is referred to as Cas9 protein. Is one of them. There are others. Now, I don't know a huge amount about this because it's a long time since I've studied all this stuff. But essentially, what you would have is your non-mutated bit of RNA, DNA, RNA. RNA is another type of DNA, essentially. So this is the sequence you want. And then this curved structure, the orange strand is one strand of your DNA, and it has an error. There's the mutation. What happens is this molecule compares the sequence you give it, because we've learned how to manipulate this now, and goes around your cell, every cell that you give it to, and corrects the mistake. It inserts the new sequence in the area where the mutation was and corrects it. And this was a big breakthrough maybe 15, 20 years ago. This was a method by which you could probably replace half of the mutations that you wanted to in a plate of cells. But still, this is done mostly in the laboratory, not really in whole organisms. I don't know if I discussed that. Yeah, okay. Yeah, so the drawback of this is this has to be highly accurate. If this same strand goes around and finds other sequences that look the same, then it will attempt to repair a strand that isn't mutated. So there's a potential for actually introducing problems if you don't manage to control this sequence of events properly. And that's indeed what we seem now to be able to do really quite well. The trouble is to use it in patients, it has to work really, it has to be safe, which means you cannot introduce any potentially damaging mutation. So it's taken time to develop this into a safe system, and I'm not sure we're quite there yet, but it is coming along and happening. So we can repair DNA mutations using this on the laboratory bench. If we combine that ability to correct DNA mutations with stem cells, so we go into a stem cell and correct a mutation. So you would get your patient, like your aspergillosis patient, take some blood, or take a piece of lung tissue perhaps, and from that you will be able to isolate the lung stem cell. And it's quite hard to do. Again, that has taken decades to be able to learn how to do that reliably, but now we can do that. Your lungs are full of stem cells which are going around your lungs all the time, repairing damage. For example, if you've had the flu, your lungs can be quite badly damaged, but the stem cells go around repairing the damage. That's what they're designed to do. If we could take the stem cells from an aspergillosis person's body that contains a mutation that leaves them vulnerable to infection, use gene editing to correct that mutation, and then reintroduce it to the person who has that mutation, could the new stem cells be relied upon to replace the old stem cells. Now, there are problems with stem cells. I think this is subject to what we talked about some time ago. We said stem cells have this marvelous potential to replace damaged tissue if we can control them. The trouble is, we've spent a long time learning how to isolate them and grow them. And when there have been experiments where they've been injected into an area, I presume this is in animals, in animal models, animal experiments, and you inject hoping that those stem cells will now repair something, it's quite hard to control where they end up in the body, and they will carry on growing no matter where they end up. And what was happening was you'd get these sort of stem cell tumors in the wrong place. So it was immediately realized that this could be dangerous, obviously. So we've had to learn to control this hazardous potential, again, to make the use of stem cells perfectly safe for people. And this is where a lot of work has been done in animal models. And it's been a big holdup. Like I said earlier, the DNA editing isn't 100% effective either. And in the paper that we're talking about, roughly half of the cells that were looked at had a repaired DNA mutation. So is half enough? Or is it enough to correct the problems caused by the mutation? Now this, yeah, okay. Now this paper, which is the paper I'm talking about, was in Science. So this is a major, one of the best journals in the world. So very reliable, very high quality. Has managed to do both in mice, which is a huge step forward. It's a group who are based in Texas, in America, and they've managed to combine these two things with a rather clever bit added on. They've combined stem cells with gene editing. It's been done in mice, and the particular mutation that they're looking at is the cystic fibrosis mutation that you can actually buy mice who have this mutation already in their lungs, well, throughout their bodies. And they've been able to isolate some, no, that's wrong. They bypassed the problem of having to isolate stem cells. They were able to treat the mice directly by injection of a particular design of nanoparticle, which carried the DNA editing system inside the particle. It was delivered, so this DNA editing is referred to as CRISPR, which you may well have heard of talk about on sciencey programs. These nanoparticles are injected, and they were specifically, what normally happens to nanoparticles if you do this, is they end up in the liver, and we didn't want them to end up in the liver. We wanted them in the lung cells, and they've been able to add in a tag on every nanoparticle that directs them into lung cells. So you can now overcome the problem of isolating cells and getting them back in and controlling how they behave by just leaving the cells where they should be and delivering the DNA editing mechanism directly to the lung cells while via injection. And they were very successful at infecting, getting this into all the cells in the lungs of these mice. Approximately 70% of the lung stem cells were safely edited. And what they actually took some of these cells out to test, because obviously they're not human cells. You can also buy human lung cells that have the same mutation. They corrected the mutation, and they carried out tests with the new Trikafta medication, which is the medication they were talking about earlier, which is a set of 3 which corrects the mutation almost chemically. And the result they got with these stem cells that have been corrected was equivalent to how well the Trikafta medications were working in this. So it's as good as something that is causing huge improvements in CF patients right now all across the world. So tremendously encouraging that this is possible. Just a word on the nanoparticle that I just mentioned in passing. This too is a huge development. Being able to direct nanoparticles to particular organs is a tremendous step forward, and it was actually carried out by the same group in Texas, and was published in 2020, I think it was. So yeah, there we go. And this is in Nature, so this is another high quality paper. So this was published in 2020, and 4 years later they published this editing of mutated stem cells paper. They then followed the mice to see if the mice felt better, because these were mice essentially with cystic fibrosis. And they also looked at how the cells survived in those mice. And what they found was the corrected cells, stem cells, and all the cells produced by the stem cells, so all the different cell types, survived, persisted, grew, spread out across the lungs, effectively causing a partial replacement of the mutated tissue. This means they've successfully got this treatment into stem cells. If they hadn't managed to get it into stem cells, this would happen, which is all the corrected cells gradually disappeared because they're recycled along with everything else. It's only the stem cells that stay there and produce the new cells to replace these other cells. So this is an extraordinary achievement. Now this technology exists. It could theoretically work in people. All this work has been done in mice. If you can think of any genetic mutation, not only in lung cells, it just depends where we can direct these nanoparticles. It could be anywhere else. I think you might have trouble getting them into brain tissue, but apart from that, this is potentially a way of correcting genetic mutations in people who've inherited genetic mutations, which is an extraordinary achievement. This is a huge step forward. This is potentially something that could be used in the future to correct the mutations that are causing your chronic aspergillosis. It's a big step now to take this technology into clinical trials. I didn't look, but mostly by the time something's been reported, and this paper was just reported this last week, they're already working on the next step. So I will keep an eye on whether clinical trials are happening with this technology. They might not be happening yet because you can imagine if you're going in and changing people's DNA, there's lots of regulation in the developed world anyway about those experiments happening. So I can imagine that being a nightmare to get through. But it's so important that you can only imagine this or some version of this will eventually be tested in human subjects. We've covered so much ground to get here. We can only hope it happens quite soon, and it will take a step-by-step approach to doing that. What is the relevance to aspergillosis? Theoretically, if we manage to get these changed stem cells into your lungs, for example, could most of your lungs become non-vulnerable to infection? That's possible. If something like you've got something like ABPA, then those cells are reacting in the ways that was described by MFIG. You could stop that happening, so that might help ABPA. However, where there's permanent damage, so scarring and holes in your lungs and that type of thing, I don't believe it will have the potential yet to repair extensive damage, but certainly there might be some benefit by correcting the mutation in large parts of your lungs anyway. But the authors of this paper would know a lot more about that than me. And that's it. That is an amazing advance in genetic disease, and it may have an impact at some point in terms of treatment, whereas the actual mutations themselves will have a much sooner impact as we try and screen out people who are vulnerable. And because I've done a little bit of this sort of work, I thought, wow, that's interesting. But I'm not going to go into any level of detail here because it would take the rest of the afternoon. So some of these things you just have to take it as read. Essentially, one of the things that this group in Nottingham is doing, and I forget the poor bloke's name now. I wonder if Anna knows, can remember.
I wonder if it might be someone I might have mentioned.
Knox, that was his name.
That might be him.
So the group
Scottish
led by him.
bloke.
They work in Nottingham, and there's a big asthma team in Nottingham, and there's another one in Leicester, I think. They look at asthma, and for some time we have known that many cases of asthma are caused once again by genetic problems. So you're genetically predisposed to get this asthma, and that to get asthma or severe asthma. We know that as a general principle, but what we actually find when you look for people who have these genes, is some people might have the genes that are supposed to predispose them to asthma, but don't have asthma, which is unexplained. Other people don't have those genes, or have genes that are supposed to protect them from asthma, because we know about those as well, but they still get asthma. So it's as if it obscures the function of the importance of these genes, because sometimes they don't seem to work properly. Why might that be? So this group did a relatively simple series of tests. I used to do something similar, but not anything like on this scale. We know that there are a series of environmental factors, and I've got another slide about that in a minute. So not only do you have to have the right genes or lack the right protection genes, they might also need a kick from the environment, and I'll talk about that. How do we understand it? Normally genes are controlled very carefully by your body. We wouldn't exist without our bodies being able to turn our genes on and off, and they can rapidly change your genes multiple times a day. Some genes are turned off or on depending on what they need to do. However, we're talking about genes being switched off permanently, or whole groups of genes being turned off permanently. And that is caused by something known as epigenetic change. It means genetic on top of genetic, so outside of genetic, and that's caused by your DNA, or parts of your DNA can become modified by something called methylation. Once you've got something methylated, it shuts down all the genes on that bit of DNA. We know, I think I've got another list coming up. We know that what causes this methylation can be anything from the environmental factors, including your diet can cause certain genes to be shut down by methylation, smoking, even aging, and there's a few more. So maybe this mechanism of large-scale shutdown, of more or less permanent shutdown of groups of genes, may have something to do with asthma. We didn't know that before now, because to look for methylation modifications is quite tricky, and the technology to do that is not commonly used. Yeah, so similar pictures to the one we saw last time. There's your X chromosome. If you tease out a strand of DNA, it's all coiled together, coils upon coils, and you end up, if you open up the DNA to the DNA helix, then literally modifications that are attached directly to DNA. and they alter the structure of the DNA. So that this DNA now cannot be opened up and used to express the gene that might be in that piece of DNA. Things that cause this modification can happen as we develop in utero or in childhood. So it's happening while we develop for some people. And sometimes that's perfectly normal and what we need to do, what our bodies need to do. Some sets of genes we only need when we're little, and we can safely shut them down as we get older. I think one example was something like lactose use used to be something that's only expressed in young babies because, of course, they need to digest milk, where the lactose comes from. And once you had grown up and you no longer used milk, you didn't need that gene, so it was switched off permanently, and it would have used methylation to do that because they can sort of switch it off and forget it. So it's kind of putting it into archive. Incidentally, one of the reasons why we can drink cow's milk now is that development switch was turned off for most people in the parts of the world that drink animals' milk. And that's the only reason why we can get benefit from drinking milk. So the system isn't can be modified. Environmental chemicals, so pollution, essentially we're talking about here, or smoking or whatever. You inhale that stuff, then it can trigger these mechanisms and shut down large parts of your genes. We don't know how controlled that is, so this is just another reason to be very careful about what you inhale, especially if you happen to be fighting fires and things like that. Then you're running a risk of being exposed to these chemicals, so you have to be very careful. Some of these things are drugs that can cause methylation. We know a bit more about that, but we don't know exactly what how much it happens with those drugs, but we can stimulate methylation using drugs, and we can also stimulate demethylation using different types of drugs. Aging and diet, we've already talked about. As a result of these mechanisms and genes being switched off, you can end up with things like these health endpoints: cancer, in autoimmune diseases, medical disorders, and diabetes are options. So this is really quite an important area of research, which doesn't, or certainly when I was working on it, didn't get a lot of mention. We didn't do an awful lot of it. It was just a nuisance, actually. It used to mess up your results, having methylation on your DNA, but I won't go into that. The research group in Nottingham looked at DNA methylation in airway smooth muscle cells. So those are the cells that constrict to make you breathless when you're asthmatic. In asthmatics, and then compared them with people who had the same cells but are people who are not asthmatic. They discovered that the DNA in asthmatics was far more methylated. So they've been exposed to something, or maybe they're just genetically predisposed to have more methylated DNA compared with non-asthmatics. So asthmatics have access to fewer active genes because of this. And could this be one of the things contributing to asthma? Never been considered before. This study is pointing the finger at maybe there are a whole set of genes that have been turned off, that if we can turn them back on again, and we can do with certain drugs, although the ones I'm aware of you wouldn't be able to give to a person because they'd be quite dangerous, because they would turn genes on all over your body, which in an uncontrolled way. But there are ways to control that being developed. And that's one of the major causes of asthma. That could be the reason. It's a simple experiment, but it might be pointing the way to some really major results. Much more work to be done as ever, but just showing you that different methods of looking at things can produce some really encouraging results and really point the way. We've learned a lot about genetics, but maybe it's about the methylation just as much as about the genes themselves. Have I finished?
Graham, is that kind of study undertaken because the numbers of asthmatics are rising at such a rate? Is that
It will be one reason we need to find out what's causing asthma. It's such a severe disease; it ruins people's lives. So this is, like I say, probably a relatively cheap experiment to do. It wouldn't be done in the same way I used to do it, and probably no one's looked before. But they've just shown a fairly basic thing, that DNA is more methylated if you've got asthma, and that's quite not exactly a surprising, but a very informative general principle that we can now use to guide and also interpret into things we already know about. So it's very encouraging. So if just knowing this bit, maybe it's been an exposure to something environmental that's caused this. If we can find out what that is, we can stop people being exposed. And that may be why you get asthma exacerbations when you're an asthmatic. It might be you're being exposed to something environmental. It's messed about with the methylation in some of your genes, and you've ended up feeling very poorly. It is possible to control, and maybe new drugs are going to be developed. And that's it.
Well, thanks for the invitation. My name is Norman van Rhijn. I am a research fellow at the University of Manchester. So I'm in the Manchester Fungal Infection Group, but partially shared with microbial evolution. And I thought today I'm going to give a brief overview of what we've been doing in terms of our research and how we're trying to impact policies to actually make a real world difference. Oh, are we going to do this? Yay. Well, I don't really have to tell you much about fungi, just a couple of things that we always think about. Fungi are incredibly useful, kingdom with many faces. A lot of these we love to eat, love to see. Some of those are minor inconvenience, but a lot of these are obviously really, really terrible. And that's the spectrum that I tend to work in, a lot of different fungal diseases, including aspergillosis. Again, I don't really have to go over this, but I've just put it there. But what's been a real great advancement for us is that the WHO has now recognized Aspergillus fumigatus as a critical group that we need to have a look at. So from a World Health perspective, we now actually have some leverage to go to the government and say, well, this has been recognized, this is a problem, so actually let's do something about it. Now, the other thing that we're always trying to think of is we think as a fungal infection group a lot about fungal diseases. But fungal diseases actually also include things like plant diseases, things that affect our crops. And interestingly, this is per weight where the most antifungals / fungicides are being used. So there is a big environmental impact of antifungal use in the environment. As you can see on the right, this is in tons. This is what's being used every year in the world. And this also includes things like the triazoles, so things that are used for treatment and are very important, are also used in the environment to treat crops. But what's important to realize here is that it's not just goes to our crops. If you spray something on a field, that also goes into the soil. And this is where things that grow in the soil, i.e. our fungi, are already exposed to these compounds, and that's where most of the resistance, at least that we see in the clinic nowadays, is being developed, with the exception of long-term treatment of mostly chronic pulmonary aspergillosis, where you see resistance development over time as well. That tends to be a completely different mechanism. Now, we started to look at fungicide policy in terms of what is required for for industry to show that a fungicide is safe to use in the environment. Now, these are sort of the most 4 categories that industrial agrochemical producers have to look at. So toxicity to things like bees, which are protected, runoff area into waters impacting fish, things like, does the fungicide impact our flowers, and is there direct impact to humans? So is it actually toxic or carcinogenic? Within the policy, there's absolutely nothing about resistance development of pathogens. So this is what we're trying to change. This all started with an example of Aspergillus fumigatus. I mean, I'll tell you about it, but the important bit here is that it's an environmental mold. So we see it in the environment, it grows in that soil, that's where it's exposed. Again, big burden of disease, and that's what we're trying to address. These are the numbers that we tend to put out to the government and say, this is a big problem globally. How about we do something about it? And we can do something about it. Now, traditionally, we've been looking at the azoles for about, or myself, for about 10 years now. And these are our first-line treatment, so things like itraconazole, voriconazole, posaconazole, isofluconazole now, to treat patients in the hospital like yourself. But at the same time, there are agricultural azoles. These have been used since the 70s as well, things like trifimazole, imazalil. These are sprayed by the tons, and this is actually where we believe that Aspergillus fumigatus develops most of its resistance. Now we see that accumulate in environmental hotspots. I'm not going to talk about hotspots today, but the most important bit is we get an accumulating antifungal resistance due to the environmental use of azoles. We highlighted that a couple of years ago when we had a look at Aspergillus fumigatus in the UK, and we got about 300 samples from across the UK. We genome sequenced them, and we found that there was a clear population structure in terms of this sort of purple cloud on the right, where we see resistance develop. So we thought there must be some sort of genetic basis within the fungus to develop resistance, and we followed that up. Now, this is some recent work from the lab where we actually looked at what is that driver. And we saw that resistance, as I told you previously, was mainly into this bright side of the clade, that purple cloud that I showed you here, sort of depicted at the top in red, and orange is these resistant isolates. When we looked into the genomes, we found that these strains actually, almost all of them have a gene, have a mutation in a gene called MHS6. And what that's important for is DNA repair. So we thought there might be a case that these strains actually have a problem with their DNA repair, and therefore they mutate much faster and able to gain resistance. Now, a lot of experiments cut short. We tested this in the lab, and actually, yes, these strains that are already resistant can develop resistance to novel antifungals much faster. So we think that there is a genetic basis of resistance development already in the environment out there. So that's what I already told you, just put here in text as well, that resistance is more likely to emerge within the organisms that are already multi-azole resistant. Now, we've been talking about the azoles. What's important to know is that Gaffey and David Denning already put out a letter to the WHO to talk about azole use in agriculture. This was in 2018. And what they came back with is, well, sorry, but the azoles have been used for over 40 years now. You're a bit late to the party. So it's probably already too late to talk about the azoles. So we've been thinking about novel classes of antifungals. There have been no novel classes of antifungals since 2001 when the echinocandins came in, which is because it's incredibly difficult to develop an antifungal because fungi are eukaryotes. They look like humans quite a lot. It takes time, and there's a considerable investment. And for pharmas, we always hear there's a pretty low return, although there are some schemes out there now which hopefully will try and change that. Now, there are a couple of new ones that are coming out, hopefully pretty soon. The one we're interested in is olorofim. Olorofim is a novel antifungal with a new target, looks very promising for things like aspergillosis and things like the dimorphic fungi and other difficult Lamentospora. This has been developed by a company called F2G, which sits here in Manchester. So for us, that was an easy collaboration to develop. Now, we've been interested in resistance to olorofim for a while, but what we found out last year, actually, that at the same time as F2G is developing olorofim, a company is developing an agrochemical called iflufenoquin with the same mode of action as olorofim. So we thought there's probably going to be a similar issue of what we've seen with the azoles with this novel class of compounds already before it's even being used. And because we had that comment from the WHO to say, oh, you were too late, we thought, let's get there first now, before it is out, because it went through the approval process, before it's being used, let's do the science and actually come up with some clear evidence that this is going to be a problem before they're even used, and take that to the government. So this is why I told you F2G started in 1998. So again, 20 years of development for this one compound. Hopefully it will be deployed next year now. But iflufenoquine, new fungicide to protect crops from fungal diseases. It's been developed by Nippon Soda, has already been approved in the US, and they're actually spraying that for the first time this season. Luckily in very limited environments, but we're actually live monitoring that to see if we get environmental resistance development. But they're still seeking approval elsewhere, like in the EU and the UK. Now, we did some work on both of these compounds, and we actually looked, is there going to be cross-resistance? And the first thing we did is some computational analysis here in the lab, where we looked at the target of olorofin and iflufenoquine. Can we, if we actually do some sort of mathematical modeling, can we actually see if these molecules have a sort of a same binding? Now, as you can see here on the left, in orange is olorofin that binds to DHODH, the target molecule, and purple is iflufenoquine, and I would say that is pretty similar. So we took this forward and actually did the lab work. And what we can do, we developed this 2 years ago. What we can do is a sort of microevolution experiment. Is you grow up a load of fungus in the lab, and during replication, you get spontaneous mutations in its genome. That means that at the end of your growth cycle, you have a diverse population of fungus that all looks slightly different. And what we can do is we can select for resistant mutants, and we can isolate these resistant populations and then look at what is different in the genome. Now, we did this, and we found 3 different mutations that conferred resistance to iflufenoquine in these 3 sites, just around that sort of binding pocket that we already identified. And when we do the experiment, we see that if we make these mutations in a strain, we reconstruct them again, we see resistance to iflufenoquine, and really scary, but not unsurprisingly, we see resistance to olorofin as well. And we can validate this on a protein level instead of doing it in a whole fungus to actually prove, yes, it is the changes in the protein that confer resistance to both of these molecules. So what we've shown then, and we published this recently, is there is going to be cross-resistance. If you evolve to become resistant to iflufenoquine, you will be resistant to olorofin from a fungal perspective. But they don't involve the same mutations, so we feel that there is a real key to studying what happens in the environment and how does that impact clinical outcome. So if we evolve a strain to olorofin, we get mutations in this G119 spot of the protein. Well, if we evolve to iflufenoquine, you get these other ones. So again, there's a clear bias of what we're probably going to see due to long-term use of olorofin and due to environmental acquisition of resistance. Now, there's no risk assessment for the environmental development of AMR. Obviously, the big challenge is the willingness of the industry to address this problem. Pharma wants to make money, agrochemical developers want to make money, but we need them both on board for a healthier world. And we need to develop tools to do this environmental surveillance to see where is resistance going to develop and what can we do about it. Now, there are some clear opportunities. So there are different inhibitors. Iflufenoquine is different from olorofin, same target, but unrelated resistance profile. So we believe there's a clear opportunity there as well for agrochemical developers and pharma to use the same target, but make sure that there's no cross-resistance when they become resistant to one or the other molecule. And we really need to make informed decisions on AMR from a One Health perspective. We need to appreciate that these fungi are living in the environment, and what we do to the environment actually will make an impact on health of people. Now, that was a lot of what we did in the lab. Then we took it with policy at Manchester. We took it to the governments. And actually, the response was pretty all right. So we had 3 discussions now. We've had it in the House of Lords, and Baroness Bennett of the Green Party followed this up and is really passionate about this. So I would recommend people writing to her as well, because she's really fabulous when it comes to these issues. We had a debate as well in the House of Commons about the dangers of fungi and what's happening in the environment. So hopefully within the next couple of years, I can update you on that. And from the other side, we've had a side of the pond, we've had some interest as well. So the Environmental Protection Agency in the US picked this up as well to actually develop a legal framework to assess the risk of antibacterials and antifungal pesticides in the environment to our health. And they've been pretty positive with the response so far. We put a letter in, and more recently, actually, the Australian government reached out as well. They're probably going to go down a different route. They're probably going to ban ipsifenequine completely. Now, that leaves me to thank everyone that's worked on this, all colleagues and friends, everyone in the Manchester Fungal Infection Group, and the Wellcome Trust for funding this work. I'm happy to take any questions.
Can you, sorry, Norman, could you say again what you said the Australians have done?
So they're thinking about, so what happens when an agrochemical, a new mode of action is going through the approval process, the developer applies to the government to say, this is the safety profile, this is what we fit, can we use it? And then the government has to approve it. While that happens, public documents being put out for people to comment on. It goes to committees that look at the safety profile and what it does. And actually, through our work, the sort of the committee that goes over that reached out to us to say, well, what's the data showing? We told them what the data is showing. Obviously, as scientists, I can show them the data. I can't make any recommendations. But their recommendation was, well, if we don't really know, how about we just completely ban it?
And did you, Norman, did you say that the majority of resistance found in patients comes from the environment rather than being generated internally?
Yes, that's correct. So in terms of, we believe, at least that is the case, because when we see mutations in the fungus due to long-term exposure to azoles, these tend to be point mutations in the CYP51A gene, so the target of the azoles. That carries a fitness cost for the fungus. So actually, it's not beneficial for the fungus. However, these environmentally acquired mutations, which is a tandem duplication in the promoter plus a point mutation, there's zero fitness cost in terms of being able to grow in soils, but also in being able to infect. So quite a bit of work has been done in the Netherlands, and they see that at least, I think it's 65% of the patients that develop resistance, it's probably due to acquisition from fungus of the environment, either replacing what's already in the lung or having resistance from day one.
I think Paul mentioned a year or two ago, he thought the average CPA patient was only infected once.
I think he's come back to that from actually more work that has been done. So it's very complex. What other researchers have shown as well, mostly Ali Reza from, I have to say this right, from King's College, he's shown that actually most of the samples he got, he actually looked at the genetic variability. So he plated out everything that he found and he sequenced everything. All of his patients had at least 3 different strains in their lungs. So if we're thinking, and that's another line of work that we're doing, if we're thinking about how does resistance develop and what's the impact, we're probably actually, most of the clinics, you grow something, you grab one and you have a look. That's probably not the right way to doing things. So
yeah.
Big push there as well.
Yeah. Interesting.
Anybody else got anything?
Can I ask a simple practical question? So, because it seems to be genetic, obviously asthma is genetic, and it's, you know, and it can end up getting asthma. So should we not allow children, grandchildren to play in the soil? Do you see what I'm thinking?
Mm-hmm.
On a simple level.
No, I completely get that, and that's a very good question that we straight away asked as well. What we currently don't know, although I have a little bit of data that we actually we only got last week, is that it does seem to be that at least Aspergillus fumigatus grows very rapidly in compost heaps and in compost bags. It does not grow so rapidly in normal soils. So my feeling is normal soil is probably fine, is healthy. Now, compost and how we make compost, industrial compost, is technically a man-made substance. So I would say that's probably a bit of a, you know, an artificial kind of environment that we're just generating. On that note, compost in the garden is different than industrial compost. So we've also looked at garden compost. That does not tend to be a problem. It is really industrial compost where they turn it, they heat it up to make it go really fast. That's where we see the fungus grow really rapidly.
Right.
That's a really interesting point, that, because we know that that's something that a lot of the patients, common theme among patients in these groups is that they enjoy gardening, and we know that a lot of them are incredibly concerned with gardening. So that's probably I imagine that's quite a reassuring point for many of you.
So I would say gardening, definitely in the garden, is great. We have sampled compost bags. They do, from industrial composters, they do contain a big amount of Aspergillus fumigatus spores. So obviously I can't, well, I'm just going to say personally then, I would not recommend going with commercial compost bags. And we've also seen that in things that are labeled, again, this is something that we're following up, but when it's labeled organic compost, that does not mean there is no chemicals in there. Organic composts mean they don't add things, but from the input that they use, anything can be in there. And most fungicides actually last longer, so they go through the commercial composting cycle and they actually just stick around. So even if you think I'm using compost and I'm putting them in my plants, you're putting fungicides straight into your plant soil.
Right. When you said garden compost is okay, my husband's got like 3 compost bins.
Mm-hmm.
2 at the allotment, 1 here. I have nothing to do with them. I go nowhere near.
Mm-hmm.
So are they relatively safe?
Yes, I would say so. We have sampled them, and it really comes from the input. So it's a combination of the input and the temperature. So the input is very much, if your input is clean, so you use your grass clippings, your vegetables, things like that, that you know are no fungicides are being used, then you have no fungicides in your compost. Your garden compost will not reach the temperatures that are required for Aspergillus fumigatus to really thrive. That is really a commercial composting process that happens. Now, I would say still, you know, we're still following this up. There is some growth in these compost bins, so I can't say they're 100% safe. I would not like to say that, but obviously, as a scientist, I'm always on the urge of caution with recommendations. But I don't think they are as big of a threat as commercial compost.
Okay. Even, this is probably silly, even if you put moldy, excuse me, moldy fruit in the garden compost, but it's still not making it unsafe.
Yes, well,
so
that is, if it's fruit, and this goes again back to the input, if it's fruit that you've grown yourself, probably fine. But if it's fruit that you've store-bought, a lot of fruit has a fungicide coating on the outside. So oranges, so things like that, a great example is oranges. And I would say when you go to the store, pick up a bag of oranges. A lot of them say what has been used, imazalil or tebuconazole. And they're used on the outside just to keep them clean.
Ah, okay. This is amazing. Okay. So generally speaking, we shouldn't put moldy fruit in the compost bin because most of it is things we haven't grown at home.
Some fruits are fine. We're actually doing, so I've got a student now who's just going to the store and buying a whole load of fruit and looking on the label, and then we're checking chemically what is on it. The big, I would say oranges are the big contributor.
Right.
Apples tended to be fine, but that's also because most people wash them and you eat them, so the peel is okay. Bananas tend to be fine somehow. I don't think they use fungicides on those, although bananas have really weird fungal diseases, so that's something really strange going on there. Big antifungal users as well are for almonds, and I'm missing one out, but I can't remember. But generally, things that sort of are more soft fruits, absolutely fine.
Oh, okay. Okay.
Can
Okay.
I ask, what about grapes?
Grapes tend to be fine as well. I would consider that as soft fruit as well. They tend to not be sprayed as much because that the spray itself will just make the fruit shrivel up and you can't use it anymore. But always, I would say always check the labels. Interestingly, if you buy organic oranges, no fungicides are being used, so they should be fine.
And would it not matter if you, like everyone's suggesting, washing everything in baking soda and vinegar and stuff, which we kind of do if we can't buy organic and we know it's all right, has whatever they've used would have gone into the orange presumably anyway, so it wouldn't matter.
Actually, it stays on the
skin.
on the outside very much. We've not. That's actually a really good idea. Maybe I should do that in the lab, wash it, and see what levels we get then.
Yeah.
Yeah, I like that idea. I'll give
It'd
that a
be
go.
interesting.
See if what we get raw makes a difference if we wash it with water or vinegar, and see what comes up.
Yeah.